Welcome to class of high throughput data generation and analysis. Dear students, today we are going to learn image processing of microarrays. On the last hour we have discussed about what is microarray and what are the different types of microarrays data, and what are the different types of the error associated with the microarrays, and how we select the microarrays sports for the further analysis. So in this class today we will study about how the image processing is done and what we need to take care of about the different issues. Image processing involves the following steps. There are mainly 3 steps. The first one is identification of spots and distinguishing them from spurious signals. So in the last class also I have told you how we should eradicate we have to discard the spurious spots and defective spots. So there are the different sources of error: contamination and beam positioning, and due to the other issues and the density issues. So the spots may be defective and they may not be in the similar format. So we have to discard the spots that are not in the same format, that are not homogeneous, and so we have to discard those spots. So now today we will learn this method how we can discard them. Okay. We need to identify what are the defective spots. You know what are the defective spots because in the last class you have learned. Now today we will learn how we can discard. What is the method? Second important thing is determination of the spot area to be surveyed. So we need to identify the area that we should consider for the analysis. Determination of the local region to estimate background hybridization, because due to the background intensity also there may be the problem in the analysis. So we have to eradicate this effect. We have to solve this problem. So spot area is important. Okay, how much area should be considered for the analysis? So wherever laser beam is positioned on the spots, so exactly spot area should be considered. So it is about the healthy spots. Next one is what is the third important thing? Reporting summary statistics and assigning spot intensity after subtracting from background intensity. Okay, so to eradicate the effect of the background intensity, we will subtract the background intensity. Okay, so in this step the statistical method is used. So these are normal methods. So this normal calculation is done for the selection of the spots. Okay, now intensity signal. What is intensity signal? So most approach use the spot median values with the background median value subtracted from it. So as the metric to represent spot intensity. What is spot intensity? Means spot median value, okay, with the background median value subtracted from it. So this can be considered as spot intensity. The median intensity is a value where the half measured pixels have intensity is greater than this value. So the spots are considered which have the measured pixels that have the greater intensity than this value. So that is called median intensity value, and the other half of the measured pixels have intensities less than this value. Okay, so that is the median intensity value. This is important in the analysis. Okay, we will see in the formula. The other method is to use the total intensity value, which has an advantage of being insensitive to misidentification of spots. So in this method, in the total intensity method, so this is the there is the advantage. So this is insensitive to the misidentification of the spores, but few more pixels with the zero value in the background will not affect the total intensity. Okay, so there may be the minor differences, but that are not important. So we can consider, we can take this method also. So this is also important, but has the this also has the disadvantage of being prone to be skewed by a few pixels with the extreme intensity value. So it may happen the few pixels have the extreme intensity value, so that may be discarded. Another consideration in the image processing is the number of pixels to be included for measurement in the spot image. Okay, means how many number of pixels we should include, can be included for the measurement. So for the many scanners, the default pixel size is 10 micrometer. Okay. So this means that the average spot of diameter is 200 micrometer. We'll have, means this is the area of the spots, 200 micrometer. Means this will have approximately 314 pixels. So this is the large area. Initially we used to take this size, the 200 micrometer, for the analysis. We generally contain the 314 pixels, but for the analysis, for the image processing, it is the best, it is better to use the smaller pixel size, so that the enough pixels can be sampled. So most scanners now allow the pixel size of 5 micrometer. Okay, now you can so you can divide the 200 by the 5. That means 40 for like, so how many pixels will be there? In the 200 micrometer there are the only total 314 pixels. Now you can calculate how many pixels will be there within the 5 micrometer. Okay, so the number of pixels will be reduced here. Expression ratio. So this is the important, expression ratio is important. Now we know how we should consider the spot intensity, what is the median intensity of the spots, and how we should subtract the value of the background intensity, median intensity. So we saw that the relative expression level for gene can be measured as the amount of red or green light emitted after excitation. So on the microarrays, what happened? The laser beam is positioned on the microarray spots. So after the excitation, there is the hybridization. Okay, because the hybridization has happened. Now we have to measure the intensity of that hybridization. So if there is a more hybridization, that means the spot will show the more intensity. Signal will be high. The most common metric used to relate this information is called expression ratio. What is this expression ratio? Okay, because we have two types of signal, red or green. So these are types of fluorescent dye which produce the different signals for the different kinds of spots, because we have taken the different conditions in the microarrays. So in the last classes also we have discussed about that. The microarrays is sampled in the different conditions, like so red is for one condition and green is for another condition. I have already told you, we can take the green color dye for the samples that have been grown, that have been sampled from the aerobic conditions, and we can take the red, we can color the red, we can take the red dye for the gene that is expressed in the anaerobic condition. So this is the ratio, the formula that you are, this is given here. So R or G, this means red or green. So this is the normal formula, standard formula. So TK is the ratio, intensity ratio. So that means the gene expressed in the red red condition means like anaerobic, and gene expressed, this is divided by the gene expressed in the aerobic condition, but this is the standard. Okay, so RK denote this is the test sample, and GK denote this is the gene that are expressed in the reference. So for each gene K on the array, where RK represent the spot intensity metric for the test sample. So this is RK represent means spot intensity metric for test sample, and GK represents the spot intensity metric for the reference sample. Now this is clear. We have to divide RK by GK. So what is the RK? Spot intensity. What is GK? Reference metric for reference sample. So here in the formula, because I have already mentioned you the spot median intensity minus background median intensity. So that will be the spot intensity. What is the spot intensity? So this will be coming by the, when we will subtract spot median intensity minus background median intensity. So this will give the value of spot intensity. So here in this formula, so the upper side, that will denote the spot intensity. Okay, now spot intensity of test sample, that will be divided by the spot intensity of reference sample. So how the spot intensity will be calculated? When we will divide spot median intensity, we will subtract the background median intensity from the spot median intensity. So this is the formula, spot median intensity minus background median intensity. So in that way we will get the intensity ratio, that is called TK. This is also median value. So this will be calculated in this way. It should be clear to all of you. So this is the how we get the intensity ratio. So where R median spot and R median background are the median intensity values for the spots and background respectively for the test sample. So R refer to the test sample and G refer to the reference sample. Okay, so these are the reference where you can read more about intensity ratio. Now this should be clear. So this was the all about this is the intensity ratio. This is also known as the expression ratio, RK by GK, and this is the important metric. And how we calculate this RK by GK means, how we take this RK means spot intensity. How spot intensity is calculated? This is the formula. Okay, this will give you spot median intensity minus background median intensity. Okay, so what is important here? This is the figure I will show him. This is the expression matrix. Here you can see the samples are given in the different conditions. So on the expression matrix, the spot intensities are shown. Here you can see the color intensity vary. So color you are not able to see, but you can you are able to visualize like the black ratio of the black color. Somewhere it is a white and somewhere it is gray and somewhere it is a black. So it shows the intensity. Okay, so this was all about the expression ratio. Now class is over.